Hi student, in this video you are going to learn about carbohydrate, which is the first family of the 4 families of organic chemicals that you will learn in topic 2. And as you can see in this picture, carbohydrate mostly fill our diet every day, such as bread, rice, potatoes, cake, sugar, and so on. Let's get ready. This is a very quick review. The 4 families of organic molecules are: 1. Carbohydrate. This is what we are going to learn in this video. Number 2 is nucleic acid. That is a component of DNA. And then number 3 is the protein, which is the building blocks of most of your body, including your skin, your hair, your fingernails. And the last one, lipid, which is fat. And again, I want to give you a quick review about the difference between monomer and polymer. In the previous video, I used Lego blocks as the analogy to explain monomer and polymer. Now, monomers are the single subunit or the basic subunit of a chemical compound, organic chemical compound, and polymer are the combination of those monomers to become larger molecule. Right. In carbohydrates, it also has monomer, and the monomer are mostly simple sugar, such as this. Maybe you have known about this. Yes, this is glucose, right? Other than glucose, we also know some other monomers, such as fructose, the sugar in fruits, for example. Yeah, and this monomer of carbohydrate has a specific name. So the monomer of carbohydrates are called monosaccharides. So the next time you hear the word monosaccharides, it is not something alien. Monosaccharide means monomer of carbohydrates. And the examples are glucose, fructose. Now, those combination of the monomers, like this one, of course, they are the polymers. And the polymers of carbohydrate has also specific name. We will talk about it later. And mostly the polymer of carbohydrates are complex big molecules such as starch and cellulose. Starch exists in rice, pasta, potato, and cellulose are the main component of cell wall. Again, the specific name for polymer in carbohydrate is called polysaccharide. All right. Now, however, there is another specific name for polymer of carbohydrates. So whenever there are only 2 subunits combined together, it has a specific name called disaccharide. So don't worry about this, because both disaccharide and polysaccharide are basically polymers of carbohydrate. However, there is a specific name for combination of 2 subunits called disaccharide, because there are so many sugars that are made of 2 single subunits, and that's why they have a specific name apart from polysaccharide, which is disaccharide. But both disaccharides and polysaccharides are polymers of carbohydrate. So these are the details, further details about the monomers of carbohydrate. Again, the name is monosaccharide. Okay. Now, monosaccharides are sugars that consist of single subunit, and they only contain carbon, hydrogen, oxygen, and the ratio of those atoms are 1 to 2 to 1. So, for example, if like in glucose, they have C6H12O6. Now you can see the ratio is 1 to 2 to 1 between carbon, hydrogen, and oxygen. It's always what happened in monosaccharides. That's the ratio of between the C, H, and O. Now, the examples that you can find in your Andrew Allen book is ribose, which is C5H10 and O5, and glucose, C6H12 and O6. Now, you can see in this picture, okay, the molecules model of glucose. So if you count, there will be 6 carbon, 12 hydrogen, and 6 oxygen, and this is the ribose. Okay, you can see the structure. So you can see in glucose, it is a hexagon structure, while ribose is a pentagon structure. So you can learn now monomers of carbohydrate are found as ring molecule. So these are some more details of features of monosaccharide. As I said before, they are ring molecules that consist of atoms which are carbon. So as you can see from this glucose, carbon, carbon, carbon combine together to become a ring structure, except for one specific place for oxygen. And the same thing also happens for ribose. Now, it is not written here, but those are the carbon, except for one spot belong to oxygen. Now, the carbon atoms that do not have side chain. What is side chain? These are the side chain. So you can see there's only one carbon that has side chain. Now, the rest that are not attached with the side chain has actually one hydrogen and one OH or hydroxyl group. So you can see from here, this carbon has no side chain. So that's why it has one hydrogen and one hydroxyl. The same thing also happens for glucose. This carbon has no side chain, so it attached with one hydrogen and one hydroxyl. So that's the second features of monosaccharide, right? Check, check. Now, the next one. A single hydrogen group on the carbon atom to which the side chain is attached. So let's see this carbon, all right? So this carbon has a side chain attached to it. So that's why the other hand, remember, carbon can make up to 4 different bonding. So the two bonding has been made to another carbon and the oxygen, and the third hand will create a bonding with hydrogen, while the other one will be the side chain. All right. Now let's take a look at to the last feature. Let's talk about the side chain. So it is said here, a side chain of carbon atoms with one OH and two H groups. So you can see from here, this is the side chain that consists of CH2OH, right? So carbon, one OH, and then two hydrogen. You can see from here. Now let's also see the ribose side chain. Okay, so you can see from here, right? HOCH2. Again, well, this is just the writing process. So actually, this is OH, and then one C and two H. So those are the 4 detailed features of monosaccharide. Okay, combined with the previous slide, it has 1 to 2 to 1 ratio, and then the rest are these 4 features of monosaccharide. So there is a labeling system in monosaccharide with regards to the carbon. So it's easier for the scientists. Later on you will see why the labeling has to happen. Well, scientists sometimes need to point out to certain carbon in the monosaccharide. So they create a labeling system. So there are, for example, look at to this glucose, there are 6 carbon, right? So which one is the first carbon, second, and so on? So they have created a rule. The first one, C1 or carbon number 1 is the carbon next to oxygen that has no side chain. Look at this oxygen. There are 2 carbon, this and this. You have to find the carbon that has no side chain to be labeled as C1. Can you see? Yes, this must be the C1. All right. Next, then C2, C3, and so on are the carbon atoms in the ring. So from here, you have to number to the next carbon. This is C2, C3, C4, C5, and then the side chain will be C6. All right. Now, how about the ribose? The same thing also happens here. This is the oxygen. Okay. Now there are 2 carbon attached to it. This one and this one. Which one that has no side chain? Yes, this carbon. So this carbon must be C1, C2, C3, C4, and this side chain must be C5. All right. Now, I think I have mentioned the last point. The last carbon number belongs to the carbon atom in the side chain. So again, it seems meaningless for now, but later on when you learn DNA, you will see that this labeling is very, very important, especially for high-level students. And there are some more features of monosaccharides that you have to learn. Okay, be patient. Don't worry, you'll be able to master it. Be patient, you can do it. Right, so there are some more details about monosaccharide you have to know. Now, in this case is we talk about left-handed or right-handed, and in Andrew Elliott, he used glucose as the example. Right, L, which stands for left-handed, and D is for right-handed, means, as you can see in this picture, so the right-handed means this oxygen has the carbon that has no side chain on the right side of it. However, in the left-handed, the carbon that has no side chain located on the left side of it. So, however, you don't have to worry about the L-glucose. All you have to learn in this topic is only the right-handed or the D-glucose, because in natural world there's only D-glucose. L-glucose can be only synthesized in the lab using the lab chemicals and equipment. So it is not something naturally exists. All right, that's point number one. Now the next thing, other than the L and the D, we also have another labeling for the monosaccharide, which is alpha and beta. All right, so let's read the description. Alpha is where the hydroxyl or OH group of C1 located at the bottom. So as you can see in this picture, this is alpha. You can see that the C1 is over here. Okay, the OH is located at the bottom. All right, while beta glucose is where the hydroxyl of the carbon 1 is at the top. So you can see from here, this is C1. You can see that the hydroxyl is at the same side of the side chain, which is on the top. All right, so hydroxyl group will be used in the condensation reaction to make polymers. Thus, alpha and beta will determine the shape of the polymer. So you will see the difference, why it is important to learn about the position of the hydroxyl, whether it is on the side chain together with the side chain on the top and become beta, or on the opposite of the side chain and become alpha. Yes, for now, just take that information, because as you go through this video, you will see that the C1, C2, C3, and also alpha, beta will be something very, very important. Now, so far you are done with monosaccharide. Now, let's go to the polymers of carbohydrate. And as I have explained beforehand, that the polymers of carbohydrate consists of disaccharides and polysaccharides. So disaccharides are basically pairs of monosaccharides linked together by condensation. Now in my previous video, we have talked about condensation reaction, which is reaction that makes polymers by taking out water, or the water as byproduct. Alright, so you can see from here, this is the D-glucose. And can you guess, is it alpha or beta? Yes, it's alpha. Why? Because the hydroxyl group, or the OH, located at the opposite from the side chain. All right, so there are two alpha D-glucose, all right, alpha D-glucose, and they want to make disaccharides. So what they have to do, if you can see, this is C1, okay, and then C5 is over here. So C1 of the first glucose will combine with the C5 of the second glucose. Okay, this is the first glucose and this is the second glucose. Now, the hydroxyl of the first glucose, carbon C1, right, will combine with the hydrogen of C5, and they will create water molecule. And at the end, okay, because there is still oxygen over here, right? So they will become the linkage, or we call it glycosidic linkage between the two glucose. All right. Now, other than glucose and glucose become disaccharide, there are some other disaccharides. Well, this is galactose. Don't worry, you don't have to know the details of the chemical structure of it. But this is just another example of monosaccharide, galactose. So this galactose, right, the beta galactose and beta glucose, right? How do you know it's beta? Because again, you can see that this OH or hydroxyl is on the top or together with the side chain. Now, they combine this OH, right, and H. So they will have oxygen and water will be produced as the side product. And we have lactose. So galactose plus glucose, we have lactose. Lactose is disaccharides. Well, do you know where lactose exists? Lactose, from its name, lactose is something to do with the milk. It is the sugar in the milk, okay? Next, we also have glucose combined with fructose. Fructose is a sugar, simple sugar or monosaccharide found in fruits. So this alpha glucose, all right, the alpha glucose, the OH at the bottom, all right, it will combine with the OH of the hydroxyl of the C over here, okay. So they will combine and then water will be produced. Okay, and then the oxygen will be the bridge between the two. And glucose plus fructose, we have sucrose. And sucrose is something that you have by daily basis, which is your table sugar. That's sucrose. Or the cane, yeah, sugar cane, that's sucrose. Alright, let me give you some examples of monosaccharides and disaccharides in your daily life. Right, there are three monosaccharides that I have here: glucose, fructose, and galactose. Right, so the first one is glucose. Glucose can be found easily in grapes. So that's why the Chinese characters for glucose is called 葡萄糖. Putao is basically the grapes, right? So if you eat grapes, you can get glucose. And the next one is fructose, which is again an example of simple sugar or monosaccharide, can be found the highest in apples. So if you have apples, the sweetness comes from fructose, not glucose, right? Mostly come from fructose. And then the next one is galactose. The galactose, again, it's kind of hard to find pure galactose in your food. But if you have yogurt, which is fermented milk, and milk contain lactose, all right? Lactose is disaccharide. So the lactose will be again separated into its monomer, which is galactose, right? So those three are the examples of monosaccharide. Now let's talk about disaccharide. The first disaccharide, the sweetness of milk, comes from lactose, which is the combination between glucose plus the galactose. That's lactose, right? And then how about the sucrose? If you still remember in the previous slide, sucrose is disaccharide that comes from the combination of glucose and fructose. Glucose and fructose combine to become sucrose, all right? And then the last one, maltose. Okay, maltose can be found easily in the germinating cereal seed or called malt. And maltose are actually two glucose combined together. Okay, disaccharide. So yeah, I know it's kind of rare in Indonesia, but they have maltose in outside of Indonesia for syrup or something. Right, so I think you can see how this concept are actually quite close to your daily life. Okay, now take a deep breath. You're done with monosaccharide, you're done with disaccharide, and this is the last one, polysaccharide. Polysaccharide means more than two monosaccharides combined together by, again, condensation. Now, there are some examples that you will learn in this topic for polysaccharide. The first one is cellulose. Yeah, cellulose is the constituent, the main constituent of cell wall. Now, it is made of unbranched polymer of beta-D-glucose. All right, so you can see from here, this is made of beta-D-glucose that combine together through the process of condensation. All right, And yeah, this is the main constituent of cell wall. The next one is the orientation of the glucose is alternated up and down, up and down. Now, here comes what makes it important, because later on in the class we can try, if it is not alternating, you can see the side chain, yeah, it is on the bottom and the other one is on the top and so on. If they do not alternate going up and down, they are not going to create a straight chain of polymer. You will see that this will happen in the next slide, that if it is not alternating, they will create a kind of like a helical structure. Now, meanwhile, the cellulose has to be straight, so that's why the monosaccharide, which is the glucose, beta-D-glucose, has to be alternated. And then the next one, cellulose strands are arranged in parallel with hydrogen bonds forming a cross-link to create structure called myofibril that strengthen the cell. So just imagine this, like yellow, orange thing are actually a line or the one line of the cellulose. They will combine together by creating hydrogen bond in between them. Okay, this is one cellulose, this is another cellulose, so they create such a strong bonding. So that's why cellulose cell wall is very, very strong. The second example of polysaccharide that you have to learn is starch. So starch are actually the component or the main staple diet of us as Asians. So we have potatoes, rice, cereals, and so on. Right. So starch, unlike the cellulose that are made of polymers of beta-D-glucose, this one consists of alpha-D-glucose. Right. So they are the main food storage, so that's why they become the staple diet. Okay, and starch consists of two types, amylose and amylopectin. Right, let's see the first one, which is the amylose. As you can see from here, right, the amylose has only 1.4 linkage and there is no branches. Yes, it's a straight bonding, all right, straight line. Now you can see from here, now this is C1, right, because it is the carbon belongs to the one that is next to the oxygen and has no side chain. Well, this carbon is the fourth, because this is C1, C2, C3, C4. Now the linkage between C1 and C4, the only linkage that happen in amylose. And you can see from here, as I said, they do not alternate, right? Since they don't alternate, that's why they become helical, right? And then the next one, amylopectin. Amylopectin, you can see from here, it's the same. They do not alternate. The side chain always at the top, right? Now, however, apart from the 1.4 linkage over here, you also can see that the side chain, which is the C6, make bonding with C1 of another glucose. So it means that amylopectin has branches because the side chain can be used to create more polymer over here, right? And again, because they both do not alternate, they both create helical structure. Like as you can see from here, this is amylopectin, this is helical, and then they have the branch. Here, the branch happened because of the side chain used to create another lines of polymer, right? Now, what's the difference between amylose and amylopectin? In daily life, if you know sticky rice, for example, sticky rice consists of more amylopectin. So amylopectin, if it is heated, it's kind of like gel, sticky, while amylose do not. So regular rice has more amylose, while amylopectin mostly exists in sticky rice. Now, more difference about amylose and amylopectin. Amylose is not soluble, this does not cause osmotic problem. right? While amylopectin is soluble. Then why plants bother to have amylopectin? Because branches in amylopectin make unloading of the glucose more rapidly. So it means that it's easier for them to, storing or breaking down the food storage, right? Because more points for glucose to attach or detach. And then the next one is amylose is soluble in hot water but does not form gel-like substance, right? Like regular rice. Regular rice is just, become cooked, but it is not sticky. While sticky rice, like amylopectin, in hot water, they become gel-like substance. And then that's why, as I said before, rice which is high in amylose is hard and separate from each other. But if the amylose is low and the amylopectin is high, it tends to be soft and sticky. Alright, here comes the last thing that you have to learn from polysaccharide, which is glycogen. So glycogen are actually the main glucose storage in mammalian liver and muscle. So this is for animal. Basically, they are very similar to amylopectin. However, glycogen simply has more 1.6 linkage, which is more branches. As you can see from here, the amylopectin has some branches, but glycogen has even more. All right. So this glucose will be released when the blood glucose is low, for example, between meals or when you have to do lots of activities. Again, by having more branches means it's easier to store or to use the glucose. And the good thing about glycogen, it is insoluble. So that's why it will not change the osmotic pressure. Because remember, we are the animal cells, it is not protected by cell wall. By having too much level of molecule dissolved means that the danger, the risk of having the cell to burst, okay, is high. So that's why when glycogen is insoluble, we can store food storage, but at the same time, there is no risk, okay, good thing, there is no risk of the cell to be burst, right? That's it for polysaccharide, and that's the last example of carbohydrate that you have to know. Congratulations. All right, the last one, wait, hold on. So I want you to download this JMOL in your, install it in your computer, okay, from this website, right? So if you want to try it at home, please do so, but we will do it together in the class. So you can start to create the molecular model. So in the school, we will use two things, which is the real thing, we'll call molymod. You can see the, the circle, the beads, the ball, red, blue, white, and so on. And we will create the molecule of glucose, alpha, beta, and so on. But you can also try using this JMOL to see the 3D. This is very cool application. So the molecule can be created digitally, and you can see the structure in dimensional. All right, don't forget to install it in the computer. All right, thank you so much for your time. You can do it. All right, biochemistry is not that hard. You can do it. Thank you.